Genes on the same chromosome, or linked genes, are not subject to the random organization of homologous pairs that form the law of independent assortment. Linked genes segregate together. In this diploid cell, the homologous chromosomes each have two gene positions, A and B. One chromosome is big A, big B. The homologous chromosome is small a and small b. The genotype for the organism is double heterozygous, big A, big B, little b. This organism is a product of a mating between a true breeding double homozygous dominant against a true breeding double homozygous recessive. The F1 offspring are the double heterozygous genotype shown here. When the genes A and B are linked like this, the only combination of gamete genotypes are big A, big B, and little b. We call these parental genotypes because they have the same composition as the P1 generation. However, in reality, this might appear in the gametes. Big A, little b, and little a, big B. These non-parental genotypes are called recombinants and appear in the gametes in varying proportions. They are the products of crossing over, and the frequency of the appearance of these recombinants depend on how far apart the two genes are from each other, because the further apart they are, the more likely crossing over will separate the chromosomes prior to meiosis. Chromosome mapping. The concept of crossing over is used to determine the positions of genes on a chromosome. It works like this. The further apart the genes are from one another, the length of a chromosome, the more likely they will separate when crossing over occurs prior to formation of the gametes. The frequency of the recombinant gametes can be determined by analyzing the phenotypes of the offspring. The more frequently the recombinant phenotypes appear, the further apart are the genes. The recombinant phenotypes that appear less frequently mean the genes are not as separated very often by crossing over. The frequency of crossing over in percent is calculated by dividing the number of recombinant types by the number of offspring times 100. 1% crossover frequency means the two genes are one map unit apart. A map unit is the distance between two genes if the crossover frequency is 1%. At 2%, the distance between the two genes is double, or 2 map units. 3% frequency, 3 map units, and so on. If I wanted to illustrate two genes A and B on a chromosome that are known to create offspring where the frequency of the recombinants is 6%, I would start with drawing a line to represent the length of a chromosome. Not indicated is the centromere. Not indicated either are the ends of the chromosome. What's shown is merely a portion of it that represents the length, no longer than the two genes are from each other. Next, add in the genes A and B, and 6% means 6 map units apart. It doesn't matter if it's A and B or B and then A. Either is correct. 3 genes, A, B, and C, are on the same chromosome. If the crossover frequency between A and B is 4.5% and between B and C is 10%, how far apart is gene A from C? First, draw in the line that represents the length of the chromosome that holds all 3 genes. Second, we assume the largest distance given to us in the problem is the furthest distance between any of the genes. In other words, if there are more than 2 genes, here there are 3, the others will be found between the 2 identified in the problem as being the furthest apart. So in this case, B and C will mark the outer boundaries for these 3 genes. The distance between A and B is 4.5 map units. A little math tells us that the distance between A and C genes is 5.5 map units, or gene A and C separate at crossing over 5.5% of the time. Sex-linked inheritance. We've already talked about eye color gene in fruit flies, but it turns out that this gene was the first gene to be mapped onto a specific chromosome. What's more, American geneticist Thomas Hunt Morgan discovered an unusual inheritance pattern associated with this gene. Morgan found that the inheritance of eye color was linked to the fly's gender. He discovered the gene for eye color was on the X chromosome, and like humans, female fruit flies have two X chromosomes, and male fruit flies have one X and one Y chromosome. So if a gene was located on the X chromosome, not only do the frequencies of phenotypic occurrence change between the sexes, but also males cannot be heterozygous for any X-linked trait. Assuming for the moment that eye color was controlled by the dominant allele R, the wild type, that's red color, would appear in homozygous dominant and heterozygous females, while it would only appear in males who simply carry the big R allele. The recessive phenotype, white eyes, only appear in females if they are homozygous recessive. With males, the white eye color can appear with equal frequency as the wild type eye color. Therefore, an indicator of a recessive X-linked trait is that it would appear more frequently in males than females. In this illustration of a cross between a homozygous dominant female and a white-eyed male, the F1 offspring are all wild type. Note the nomenclature for displaying sex-linked genotypes. In the F2 generation, the white eye phenotype appears only in the male offspring. People who are colorblind cannot see the number 8, shown here. Red-green color blindness, a genetic trait, affects 8% of men, but only 0.4% of women. If it was a Y-linked trait, it wouldn't appear at all in females, since they don't carry the Y chromosome. The asymmetry between the sexes suggests it's an X-linked trait, and one that is recessive. The big C is the normal allele. The recessive small c is the colorblind allele. A colorblind male does not produce any colorblind offspring, but the daughters are heterozygous. Should one of them grow up and have kids with a colorblind male, some of their offspring could be colorblind. Should one of the heterozygous offspring grow up and have kids with a normal male, see how there's a 50% chance their sons will be colorblind. We usually only talk about X-linked traits because of the larger number of genes on the X chromosome, because the X chromosome appears in males and females. If you're ever asked to determine whether a trait is X-linked or linked to an autosome, look for a disparity of incidence between the sexes. Hemophilia, for example, is an X-linked recessive trait. It appears more frequently in males than females. Congenital hypertrichosis is an X-linked dominant trait that results in abundance of body hair in males and females. A very rare condition, 55 cases since the Middle Ages, affects females significantly more frequently than males. It's worth reminding you that dominant traits are not always desirable traits and still can be very rare, despite their dominant genotypic positions. Polydactyly, extra fingers and toes, is also a dominant trait. So why are these dominant traits so rare? This question will be revisited when we discuss population genetics. There are practice problems in your McGraw-Hill textbook on pages 603.